Hello, everyone. Very happy to be here to give a very basic introduction on the atom probe tomography reconstruction. So I think for the beginners of atom probe tomography region, and also for the old users, sometimes we are curious about reconstruction protocols and what is main principle behind atom probe tomography reconstruction. So today I'm going to show some details, and I think there are so many parameters that we need to use during the reconstruction. For example, image compression factor, field evaporation parameter, and also like reconstruction protocols, dynamic reconstruction, and pose and spatial distribution map, and all delocalization parameters. For this, I have put it into the video here, and I think because of the time limit, we are not going to review some details of this one, these concepts. So today, I mainly focus on the introduction of the APT calibrator software. Meanwhile, we will also include some like principles of reconstruction. Okay, so now we have 3 warm-up questions. Question 1: for the same data, during the reconstruction of an APT dataset, if we use a smaller KF value, will we obtain a longer or shorter tape? Please explain why. Yes, you are right. The answer is shorter, because once we have a smaller KF value, that means for the same electric field, then the radius will become larger. That means during the reconstruction, we will have artifacts that the software just treat your tip as a shorter tip. And question 2: assuming you use a detection rate of 1% and a pulse rate of 200 kilohertz, how long would it take to obtain a dataset of 36 million ions? So you can use a calculator to calculate, or just simply guess. Okay? Okay, so the correct answer is 5 hours. So a detection rate of 1% means that one heating event occurs per 100 pulses. So that means if we have a 200 kilohertz pulse, that means 200,000 pulse per second, and we have one heating event per 100 pulses. That means we can divide 36 million ions by 0.01, which is the detection rate, and divide the pulse rate 200,000, and also like 3,600 for 1 hour. So we get the result of 5 hours. So now you can see for if we use a smaller pulse rate, then it needs longer time. And then the third question. So during the APT, so we know that if we use a higher detection rate, which might cause the fracture of your sample. So for example, if in the 96, so if this detection rate is 2%, That means if you reach to 2%, your sample will fracture because the voltage is too high. Then if you move your sample to 76, which is a street detector, so what will be the limit? Yeah, we have one tip. The detection efficiency on 96 is 50%, and detector efficiency in 76 is 80%. Here I gave the answer. The detector rate limit is about 3.2%. So yeah, because then if a detection rate of 2% means that 4 ions hit to the detector for efficiency, detector efficiency of 50%. Then for 76, it has a higher detector efficiency. So that means if we have the same 4 ions that hit to the detector, then we can detect 3.2 ions. So then the detection rate is 3.2%. So that means in the 76 you can use a little bit higher detection rate. Okay, then we move to the main question today. The main question is how we can achieve a precise reconstruction of an atom probe tomography dataset. And if you look in the APT software, you will find so many reconstruction protocols. For example, voltage, fixed shank, then dynamic reconstruction, tip profile. And you will see here we have a comparison between a standard reconstruction protocol which use voltage curve and a constant image compression factor and the KF factor. And here you will see a reconstruction result after using dynamic voltage protocol. Now you can see the length between them. You will find a very huge length difference. And also from here you can find that for this standard reconstruction protocol, from the beginning to the end, you will find the trend of the shape of precipitates in your tip. And this is obviously an artifact. So, but if you use dynamic voltage reconstruction, you will find from the beginning to the end, then from the top to the end to the bottom, you will find the precipitate keep spherical shape, which means the artifacts has been inhibited. So why? Okay, so we know that from many previous studies, we have reviewed that the reconstruction parameters, for example, ICF and also KF, is dependent to the shank angle, voltage, and apex radius. So we have some relationship between maybe a little bit complex between the reconstruction parameter and also the factors that affect them. And also we have a relationship between the ICF and KF. And if we simplified this relationship, we can get this KF is approximately proportional to cube of ICF. And if you also like to do some approximation, you will find that you will get a relationship between the KF and the initial KF and also the voltage curve. So if we only count the inference of voltage to the KF, so this simple equation that we adopted in the APT software is called dynamic voltage. So once you use this dynamic voltage, it actually uses this protocol. So to do this in the APT, you need to select this dynamic voltage, then check this auto checkbox, then you will get dynamic voltage result. And this is like a plus function to the voltage reconstruction. So before we only use voltage, now you can just choose dynamic voltage, then it will automatically add an correction to the reconstruction parameters. And if you want to, but here you cannot find the details of the generated KF curves or ICF regarding to the detection sequence. So actually you can install the dynamic reconstruction modules from from extensions, and also activate in your APT software. Then you can just do the customer analysis and choose this GPM, dynamic reconstruction parameter. Then you can run, you can input the voltage file, and also initial KF and then the initial ICF. Then you will have the evolution of your reconstruction parameter. Okay, so then I want to also ask how we can further increase the precision of reconstruction of an APT dataset. I think the answer may be the TEM. If we have the TEM image, we can do the correlative reconstruction between TEM and the APT to further improve the precision of reconstruction. Here I show the APT, the multifunctional software for reconstruction calibration that we have been developed for many years. So you can see in this software we have some modulars, for example, image compression factor calibration, KF factor calibration, pole figure simulator, runtime calculator, and also dynamic reconstruction. And if you open this software, you will find a very simple interface here. So for the region here, you can input the poles coordinate and also the Miller indexes, and it can calculate the ICF value. And also if you do you generate a spatial distribution map, and you can select your poles, and you will get theoretic distance between or lattice spacing, and you put an initial tip radius, and you can observe the lattice spacing. You can do the iteration of the tip radius. Then you can get the KF factor. So the basic workflow is when you do the analysis in the AP suite, you can export the EPOS file, which contains everything that the software needs to do the reconstruction. So now you input into the software, and then you get the atomic positions, and also you can in the software we provide a golden seeds function. That means you can choose feature points inside the TM figure and also the feature point in your APT data set. Then you the software will automatically match the distance between different points. That means we did a segmentation of the data and do the reconstruction between in different segment. Okay, so and also here, as I mentioned before, so we can choose the location of this pose and input here, and also use standard pole figure to obtain the Miller indexes and get the ICF value. And also here, you can use iteration of tip radius to get a better fitting of observed spacing between lattice with theoretic lattice spacing. Then you will see after 2 iterations, and you will get convert result. And also if you know the evaporation field and also the initial voltage, then you will directly get the KF value. Here I show so how we did such process in the AP suite. So now you open this Reconstruction Explorer. So you generate the spatial distribution map. Then from a pole region, now you can see the lattice spacing. So and you generate the spatial distribution map and also get the distance between the peak here and the center peak, which means lattice spacing. And you input the observed value here, and now it will generate a new tip radius. So, okay. Then you use this tip radius and you will get a new KF value. Okay, and the software also include some calibrations based on the instrument. So for example, we have the direct detector in the 76, and also we have a reflection detector reflection system in the 96. So here, by check this box, then we will automatically like rotate the detector or rotate your reference. So for example, here if you place your sample on your clip holder like this, then if you check this XS, then now it is exact coordinate that a sample is facing to you. So now you will get the match between this coordinate and this coordinate. That means if you have an EDS result, you can input or you can mount the sample like this. So then you can use the crystallographic orientation, and you can do the simulation of your pole figure. And and you can also put your sample into the 76. So here you see this is the same sample, but I run twice. So before it was in the 96, and later I transferred to the 76, and we can see we get different pole figure. That means the detector installation angle is different, and also because we have a refraction system, then we will have the mirror of the pole figure. So this function has been included in the software. So that means if you select different instrument, it will automatically calibrate the angle differences. Okay, so and also another very convenient function is, so for some samples, if you have a grain boundary inside your sample, you can use the function here, the pole simulator. So firstly, you determine the orientation of your top grain, and then you determine the orientation of the bottom grain, and you can use this function here to save the orientation of the grain 1, grain A here, and also grain B here, then calculate the misorientation between these grains. And also it will give you the misorientation angle and also axis, and also will give you a prompt whether this grain boundary is CSO grain boundary or not. And as I show here, so you can use the crystallographic orientation you obtain from the TKD and do the simulation of poles. In some cases, if you have a very few poles in your pole figure, it's really difficult to identify the pole indexes. So now you have additional information from the TKD. That means you can do the simulation and also get the right index of this pole. And also you can compare with the pole figure we obtained from the experiment. Now you can see they are closely matched. So yeah. And also if you have a EBSD result, you can also do the similar work. And for example, here you see the region, because of the existence of grain boundary, and it includes 2 grains. So we have a limited region for the pole figure for grains. So for grain 1, we only have 2 poles. So if you have a TKT or EBSD data, you will have a simulation of these poles. Now you can see, now we know this pole is 111 pole and this pole is 110 pole. And also for another grain, so you can also do this simulation, and we know this pole is 001 pole. Okay, so here I show in the software how we do this. So firstly, we have a wonderful pole figure. Then we can just capture the pole figure and load in the software. Then now you can tune these Euler angles, so to match this pole figure. now you can tune the image compression factor to obtain a better result. So now you can get a correct image compression factor and also the orientation of your sample. Okay. Then next, I want to introduce a golden seeds method. So in the software, we also provide a reconstruction method, which we call it golden seeds method. So the main principle is we use some feature points we obtain from the TEM or STEM or 40 STEM to do a correlative reconstruction between TEM and the APT result. So firstly, we obtain the TEM, then we can get the coordinates of such precipitates, and or we obtain the location of grain boundaries. So now we can have the coordinates of these seeds, and also we get the coordinate of these seeds in APT. Then we can do like the correction of KF and ICF and obtain a better reconstruction result. Here I show basic reconstruction protocols. So this is best protocols that published in 1995. But it's old reconstruction protocol, and it will generate a very big distortion of your sample, but a distortion of your reconstruction. But here we can use this protocol to review how we do this work. So from this basic equation, we can see that the delta Z is approximately proportional to the square of the ratio between the ICF, the KF and ICF. So which is a principle that we do the golden seeds method. So we measure the distance between different feature points in that direction. So then we further tune the KF and ICF to have a better reconstruction result. And in the software, instead of using a bus protocol, we use the GOAT protocols, because in the bus protocol we have a reverse projection of these ions to here, so which is just a straight reverse projection, but it will cause a distortion of your data set. So in the GOAT protocol, we use a reverse projection of the detector on the surface, spherical surface. So it will improve the reconstruction result. Okay, here I show the result when we use such golden seed method. So now we obtain the golden seeds from the stem image. You can see clearly see the edge of this phase boundary. And if you do the initial reconstruction of your APT dataset, and you don't know which KF and ICF you use, you can just randomly use a value and to generate a reconstruction result like this. So now you can just you can just rotate your sample and to match the direction of your APT, three-dimensional APT dataset to to match the direction what we view here, then set the golden seeds inside your APT datasets. So for example, you put 5 points here, so which are the locations of a grain boundary or the top of the tip. Then it will automatically calculate the distance between these points. So now you see this is the distance we measure from the TM, and also this is the distance we measure from the APT result. Now you can see the distance ratio. Then we calculate the square root of distance ratios, and then obtain a new KF value here, and also a new ICF curve here. So although you see this is a very small change of ICF, it might cause the it can give you a better reconstruction result. And as I presented here, so this is the result after dynamic reconstruction. So now you can see between the TM result and this APT result, they match very, very well. And also in the software, we also provide a function that you can hit the button here, so which can give you the result by using the Hartog protocol. So yeah, so I present here. So this is the results with dynamic voltage reconstruction. So now you can see, but the main problem of this this method, you must have a very good initial ICF and KF at the top of your data set, reconstruction data set. So which is not very applicable for many cases. So then here we use our software. We provide another possibility that you use 4D stem or APT or stem images, you will directly obtain the KF and ICF of your data set. And here I show other applications. For example, if you have a grain boundary inside your needle, so then you can also use this golden seed to do the calibration. And you will see after calibration, then you see the distance between these golden seeds are closely matched. And if you use such dynamic reconstruction from the beginning of your data set to the end of the data set, you will find, before, if we don't use this method, we will find the lattice spacing will deviate from theoretic lattice spacing. From if we further goes down to the bottom of your data set, then you can see a huge deviation here. Then if you apply the dynamic voltage or dynamic reconstruction, then you can see from the beginning to the end, so or the lattice spacing match very, very well. Okay, here I show in the software how we use the function. So here we can just open the window for doing the reconstruction. So select the pose file, then it can just give you visualization of your sample. Then we'll rotate to make this edge very clear. Although we cannot guarantee that your view direction is 100% align with the direction from the TEM, but we can. I think this is a little bit like a shortage. So now you can just set the scale bar and also the golden seed from this APT result, and also you can open the stem image and also set the golden seed in the stem image. So now you have a coordinate of this point, and you can obtain the result, and it will generate the KF and ICF curve. Okay, now you can input the EPOS file and also load the range file. Okay, set an initial KF and ICF. Or you can neglect if you have the curve here. So then, so now you can see after reconstruction, you will see a new result that is better than the result before. Okay, so you can find all the tutorials from the website here, and also you can find the open source software in the GitHub, and also you can download demo datasets for practicing this software. Okay, thank you.